Good evening, Professor Amit Basak, my esteemed colleagues, and my dear students. On behalf of the Postgraduate Department of Chemistry, Lady Brabourne College, I feel honored to welcome our distinguished speaker and well-known academician of our country, Professor Amit Basak, FNA, FASc, FNASc, FRCS, FAScT. At present, he is INSA Senior Scientist, IISER, Kolkata, and formerly he was Professor of Chemistry, IIT Kharagpur. We welcome Professor Amit Basak in our DBT-sponsored state-level webinar. Professor Basak did his PhD from Calcutta University under the supervision of Professor S.K. Talapatro, and then did his PhD, DPhil from Oxford University, UK, under the supervision of Professor Sir J.E. Baldwin, and then went to Johns Hopkins University, Baltimore, USA as postdoctoral fellow with Professor and worked with Professor Craig A. Townsend from 1987 to 1991. His broad area of specialization is organic chemistry. He is recipient of many awards and distinctions, to name a few: Sir J.C. Bose National Fellow, December 2011 to April 2020, Fellow of Indian Academy of Sciences, Bangalore, Fellow of Indian National Science Academy, Fellow of National Academy of Sciences, Fellow of West Bengal Academy of Science and Technology, Fellow of Royal Society of Chemistry. His research interests are in the areas of design and synthesis of novel indines as DNA cleaving agents, synthetic potentials of diradical generating processes, enzyme-mediated synthesis, substrate analog approach for enzyme active site identification, mimics of secondary structure of proteins, enzyme inhibition approach to drug design, drug delivery, proteomics, metabolomics, protein capture, LALDIMS, and computational chemistry, to name a few. All of us use antibiotics often for various reasons, but not always fully aware about their side effects. The most alarming one is the antibiotic resistance. Today, Professor Basak will deliver a lecture on antibiotic resistance and the never-ending battle. The students will definitely be benefited from his illuminating lecture, and I am sure all of us will enjoy this talk very much. With these few words, I request Professor Amit Basak to start his talk.
Thank you, Hemanti, for your kind introduction, quite elaborate introduction. I also thank the principal of the Lady Brabourne College and many other organizing committee members. Dr. Monisha Mukhopadhyay, Hemanti is there, Dr. Nupur Basu, and the entire team for giving me the opportunity to deliver this webinar, come seminar. Okay, in a college which is Lady Brabourne College is basically during our time, I think still now it is one of the topmost, and it will retain its glory and go further up, I am sure. But there was always a competition between our college, Presidency College, and Lady Brabourne College, that who performs better. Okay, so it was actually one of the most famous college in the entire country, not only in Bengal. And definitely it's a great opportunity for me to give a talk to the students and also the faculty colleagues on this occasion. This is a DBT sponsored program. Okay, as Dr. Mollik was rightly saying that every one of us at some point of time have used antibiotics, and there is now a notion that is in our mind that it is always available. So whenever I have some fever, I will go to the shop and then buy the antibiotic. Okay, Now, that actually has created a lot of problems, and I will try to focus on few things. First of all, what is the challenging problem today? The challenging problem is the resistance of these to these antibiotics by the bacteria. But what happened in the last 2.5 years, because of this COVID pandemic, this issue has gone a little bit in the sideline, because everybody is now worried about COVID. And but I think the major problem, once the COVID is over, I think the companies and the scientists, the pharmaceutical companies will again come back and try to take this challenge little more seriously. Okay, let me go to the full mode. So antibiotic resistance problem and the never-ending battle. That is the talk. Now, in order to understand the problem and the resistance, basically the resistance problem, one needs to know that the history little bit of the antibiotics. Like if I want to play a game, I should know about the opponent. I should know how the opponent works, how the opponent is, what is the strategy of the opponent. Okay, so that is a must. That means if I want to know the resistance of antibiotics, then I must know how the antibiotics in the first place works against the bacteria. So I have actually arranged this talk in such a way, it's like a story that before antibiotics, what was the state of affairs at that time in the global context? And then after the discovery of antibiotics, how the life has changed, and now what is the present scenario. And then we'll come to, so it will be a basically interdisciplinary kind of talk, because it will be at the interface of biology and chemistry, but I will try to, because I am also a chemist, so my attention will be at the molecular level, that how the antibiotics, what are their structures, how do they work, and all these things. Okay, so without wasting time, let me go to the first slide. The first slide is basically that what was the average life expectancy over the centuries. This is just to tell you about we should have some rough idea that how the life expectancy, which is about around 80 years today, how it has been increased. If you look at the different centuries, say like 1400 to 1700, the average life expectancy was about 35 years. This is average. Remember, there are advanced countries which are having higher life expectancy, but if you consider globally, it was around 35 years. That is the longevity. 1800 to 1900, about 50 years. So it has increased. That means people have started researching and trying to find out ways how to live longer. 1950, you will see a jump from 1950 to 2000. It is around 70 years, global life expectancy. And today it is around 80 years. Now, how this thing happened? It doesn't happen without any effort or research. So I will talk about that what happened during this early 1900, and then I will go to the 2000, and then, sorry, this is 2020. What is the status? Okay. Now, I think I can skip this. This is the Middle Age was we all know the Middle Age is also called the Dark Age. There was a lot of problems, lot of infectious diseases came in, plague was there. So life expectancy was less at that time. However, even at that time, two things were known. One was that wine was a mild antiseptic, and opium is a painkiller. These two were known. So that made a little bit the progress in the surgery of a person. Like here is a cartoon picture removing gallstone. It was so painful in those days, but some pain-killing action of opium relieved some of the pain. So this was the Middle Age. These are the two developments that took place. And during the early modern era, there were two significant developments. One was that it was proven by Harvey that blood circulates, that there is something called blood which circulates in our body. And then Leeuwenhoek's, you have heard his name, that he discovered the microscope, and through the microscope, people started observing very minute things which are difficult to see under the naked eye. Okay, so the different germs, bacteria, microbes could be seen under the microscope. So this is a landmark discovery. So once that is discovered, then people started developing different techniques to combat diseases. Okay, so just a quick brush up of the history. That 1796, so close to 1800, Edward Jenner discovered vaccine against smallpox. So before the development of any antibiotics or antibacterial agents, the smallpox vaccine was discovered by Jenner. And there was a good story. If you are interested, you can go through the story, because always, you know, the discoveries have a story behind it, every discovery. Then came Joseph Lister. He developed he's called the father of antiseptic surgery. So during surgery, there is always a chance of infection. So just by using phenol or the carbolic acid spray, he could actually prevent this infection of the wounds, okay, which is done during surgery. Then Robert Koch discovered the germs. Now, there was actually always a debate that when a person falls ill, then what happens? Is it due to his sin, that due to his some bad work, he's a bad person, or is it due to something coming from the outside, which was called the germ theory? And Robert Koch discovered that there are germs of some diseases called anthrax and TB. And then Louis Pasteur established the germ theory of disease. That means the infectious disease that we have that inflict us, that is actually coming from foreign particles, which are called germs, or the bacteria, to be specific. So that was proven by Louis Pasteur. But there was still no cure against this germ. Then there was okay, this is some of the molecules that came into the market later on. These are some of the antibiotics, but I think at this point it is not very important. Let me go to the go to this slide. I think just a second. Where is that? All right. Yes. So early 1900, what happened? There was a scientist, Paul Ehrlich. I hope you can see the, let me see. I better use the pointer, laser pointer. Yes. Paul Ehrlich, he was awarded Nobel Prize in 1908. 1901 was the first award of the Nobel Prize. And 1908, he got the Nobel Prize because he thought that there must be a way to specifically kill the germ which is infecting the body by a chemical. And this chemical he called the magic bullet. So he had a theory which is called magic bullet theory, that you can take this magic bullet from outside. Magic bullet is nothing but a chemical, and this chemical can selectively go inside the body and circulate in the blood and ultimately kill the bacteria or the germ. Okay, so he made different compounds. At that time, it was known that some of the metal compounds, the metals are very, very toxic. So in the initial days, the work was mainly concentrated on metal-oriented organic frameworks, like a compound called sulverson, or also known as arcephenamine. This is the number 606th compound that Ehrlich made. He made thousands of compounds. The number 606 was found to be having antibacterial properties. Okay. And this is finally, the structure was not known at that time. Finally, the structure of salvarsan was solved, and it was found that it was a mixture of different arsenic based organic materials. Okay, so that was the start, but this is an antibacterial. See, there is a difference between the antibiotic and antibacterial compounds, although we generally always say antibiotics. We never say antibacterials. Antibacterials are basically any compound which kills the bacteria. And there are different classes of antibacterials. One is basically which are produced by microorganisms, and they are called antibiotics, like penicillin. Some which are produced synthetically. So that is synthetic antibacterials compounds. And some are called in between, semi-synthetic, that you isolate from the natural source and do some chemistry on it, some changes, put some functional groups, and then they are called semi-synthetic antibiotics. Actually, majority of the compounds that we see today, which kills the bacteria, are semi-synthetic antibiotics. But as you know that for colloquial use, we generally call everything as antibiotic. But there is a difference. Antibacterial is the key figure, and then it is followed by divided into antibiotic, semi-synthetic antibiotic, and synthetic antibacterial compounds. Okay, so this is a synthetic antibacterial compound. These are not antibiotics. So after Paul Ehrlich, then there was a pride which is for our country, that is by Sir UN Brahmachari. So he used an antimony-based compound, which was very specific, which was called ureastibamine, which was very effective against the black fever, Kala-Azar, which is caused by leishmaniasis, a form of bacteria, okay, and with microorganism basically, and then parasite to be specific. And this is an antimony-based compounds. And as I said, that those days people knew that some of the metal compounds are having toxic effects. So they wanted to utilize this toxic effect to kill the, to use as magic bullet to kill the invading germ that is entering into the body. So after UN Brahmachari, then came in 1939, a Nobel Prize was given to Domag. He discovered a compound which is called Prontosil. Prontosil is nothing but, I think the structure is basically, this is an aromatic compound with two amines, then an azo functionality, and then a aromatic compound with a sulfonamide derivative. So this is another synthetic compound, antibacterial agent, and which was, this is well-known sulfur drug. But remember, none of these are antibiotics, and none of these actually solved the entire problem of infectious diseases caused by bacteria. Okay, so what happened in between? This is actually how these arsenic compounds, antimony compounds, and the sulfonamide compounds. This is the sulfonamide compound, Prontosil. They are actually called prodrugs. Prodrugs means they go into the body. They are not actual drugs. And then they undergo metabolism and is converted into a simpler compound. If I look at this Prontosil, this is converted into para-aminobenzene sulfonamide. Okay, these are called PABA analogs. PABA means para-aminobenzoic acid. Now, this is a constituent of a very important vitamin which is called folic acid. So this PABA analog, when the bacteria synthesizes the folic acid, or when we take folic acid from outside, this folic acid is not, this PABA is replaced by these groups, and so you are not getting folic acid. That's why these are called PABA analogs. Okay. Now let us go to the story of the antibiotics. Again, remember, these are antibacterial agents which are synthetic. These are not produced by any microorganisms. Okay. Now the real story of antibiotics started on September 3, 1928. Sir Alexander Fleming, this is his laboratory. He was working on the development of antibacterial agents, and he was checking many compounds. One of the compounds that he was investigating was present in our tears. In our tears comes from the eyes. The tears actually have a natural protection against the bacteria. Because if you look at your face now, tears, the eyes are the eyes, mouth, and the nose. These are the most exposed items to the outside world. So eye infection, there is a great probability, but the tears protect our eyes from infection. So what is present in tears? He was very enthusiastic, and he developed a compound. He actually isolated a compound from the tears, which was called lysozyme. And but it was present in such a small amount. He was trying to get it in larger amount, but he failed. So he was kind of very dejected person, because his research was not going as per his wish. But what happened, one nice day, which was September 3, 1928, he noticed that one of his plates, see, when you have this test that whether the compound has antibacterial activity or not, so you have to grow the bacteria in a plate, which are called petri dishes. And this plate, you add your compound, and if the bacteria does not grow, then you will see a very clean zone around your compound. So you know that your compound is having antibacterial agent. So he was having series of plates, as you see in his lab. And suddenly he discovered that in one of his plates, that there was something which is being produced. Now I have a little bit of a video. I hope it works. Let's see. I think I will have to come out of this. If it does not work, just tell me whether you can see can you see the slides?
Yes, we can.
Yes, okay. I have actually switched off the sound. So basically, this is a kind of a video how Alexander Fleming accidentally discovered penicillin. So Fleming was basically a son of a family which was basically a sheep farming family. But he was very curious to have education in medical field. So he came to London, studied at the St. Mary's College, got his MBBS degree at the St. Mary's Hospital, basically. This is the hospital, and this is his lab. I showed you the building. And this is the St. Mary's Hospital. This is Alexander Fleming. And he was checking different, as I said, lysozyme was one of them, but he was also checking other compounds. He was dejected, as I told you. So he went for a holiday, keeping everything very untidy. He was a very untidy person. You know, research can be very tidy. When you do titration, some people I have seen very tidy, and some people may not be that tidy when they do the titration. So actually there are both plus and minus of both this. Like in Fleming's case, when he came back from his holiday, because he was very untidy, so one of his plate, which was not very close from outside world, happened. What happened? He was checking all these plates, and he suddenly found there was a plate where there was a fungus. A fungus has grown inside, and there was a clear zone where bacteria has not grown. Usually what people actually clean all these plates after the experiment, but Fleming was dejected. He had all the plates piling up on his desk. He went to holiday, came back, and suddenly he saw this. Now you can see that he is lucky, but I will say that he has an extremely brilliant mind and brilliant eyes, that he could check all these individual plates, and he found that in one of these plates, see, this is the what ultimately turns out to be. Sorry, let me again go to that. If you just a second, yeah, this is the plate where you add the compound from the fungus fermentation medium, and he saw this clean zone, as I showed you, the clean zone again, little bit. Yes, this clean zone, that means it has got antibacterial activity. Okay, then he showed that it can kill many of the bacterial diseases like pneumonia, like syphilis, like gonorrhea, like many other diseases which are caused by bacteria, diphtheria, etc. However, the problem was Fleming immediately realized that this fungus is producing some chemical, which he named as penicillin. But he could not isolate large amount of penicillin or any penicillin in pure form. The reason may be that he was a microbiologist. He was not a chemist. So chemists know how to isolate the compounds, and especially compounds which are very unstable. So Fleming lost his interest in this compound, so he left this research. Then two gentlemen at Oxford, again I showed their faces, Howard Florey and Ernst Chain. Ernst Chain actually left Germany because of Hitler, and this man came from New Zealand, Howard Florey. So these started again. See, the World War started, and there were a lot of infections during World War because bullet injuries are there. So World War started, so there was a renewed effort. Government started renewed effort to discover the antibiotics. So these two gentlemen went to Fleming. They saw Fleming's paper, went to Fleming, and then got that fungus, which is Penicillium notatum, and then they isolated the compound. So this is the Nobel Prize winning ceremony where Fleming, this is Chain, and this is Florey. They were given the Nobel Prize in 1945, just the end of the World War. And everybody in the world thought that, yes, now we have a cure, a miraculous cure for all bacterial diseases. However, let me stop this just a second. However, what happened that during Fleming's Nobel lecture, see, everybody has to give a Nobel lecture at the Nobel Prize ceremony. So these are the three people. So what Fleming said at that time during his Nobel lecture, the last few sentences I will read that, but I would like to sound one note of warning. Penicillin is to all intents and purposes, sorry, again I will take the laser pointer. Yeah, for all intents and purposes, non-poisonous. So there was no need to worry about giving an overdose and poisoning the patient. Penicillin was very harmless to the host, that means a human being. So if you take greater amount of penicillin, that is not any danger. But there may be a danger, though, in underdosage, that it is not difficult to make microbes resistant to penicillin in the laboratory by exposing them to concentrations not sufficient to kill them. And he said that time may come when penicillin can be bought by anyone in the shops. That is the danger. Like in our country, we can buy almost any drug, any antibiotic, because it is not prohibited. Although it may be prohibited, but we have all connections. We go to the shop and we ask for any antibiotic, and that is given. Okay, but in other countries, advanced countries, antibiotic you cannot buy by yourself. It has to be prescribed by a doctor. And so the reason I said this is that in 1945, Fleming, such a brilliant person, that he realized that maybe one day will come that penicillin, because of this underdosage and overuse, misuse, everything, that it may become ineffective later on. Now, along with these three gentlemen, later on, Dorothy Hodgkin at Oxford, he established the structure of penicillin. I have given one of the structure of penicillin. George Sheehan, he chemically synthesized this molecule at MIT. And Sir Jack Baldwin, he discovered the enzyme that makes the penicillin in the microorganism. Okay. So these are some of the developments that took place. Now let us talk about that how penicillins really kill the bacteria. Okay. How penicillins kill the bacteria. Now, if I show you by, let me see, if I write, see bacteria, you know gram-positive and gram-negative bacteria. For simplicity, we are talking about the gram-positive bacteria. See, gram-positive bacteria, what happens? They actually have what is called a cell wall. This is what is called a cell wall. And there are all this DNA, the chromosomal DNA, and then other different enzymes which are present, other metabolites which are required for the survival of the bacteria. They are inside, okay, cytosolic material. But this is the cell wall which is not present in the human cell. Okay, so there is the difference. Penicillin actually works against this biosynthesis. That means against the biosynthesis means how the bacteria makes the cell wall. Penicillin stops this cell wall biosynthesis. Now, why this cell wall is needed and not the membrane which is present in our cell? Because the inside osmotic pressure is very high. So you need, is very high. So what you need is a strong wall to protect the cytosolic material. If this wall is weak, then it will burst open and everything will come outside. And this is what is called lysis of the bacteria. In fact, the lysozyme, which I talked, that what Fleming was interested, lysozyme, which is present in our tears, that actually hydrolyzes these walls. Like we say today that soap is the best material to wash our hands when we come from outside to get rid of the COVID, which is maybe which may be present in our hand. Soap is like soap can dissolve this bacterial cell wall, the viral coat immediately. So this is the bacterial cell wall can be the synthesis of that can be stopped by this action of penicillin, because the difference between soap and this penicillin is that soap you cannot take inside in your body. You can only apply outside, but the bacteria has gone inside your body, so you cannot take a soap. So you have to have chemicals which are harmless to the host, but which are harmful to the bacteria. Now this is the how the bacteria synthesize the cell wall. Okay, the cell wall is basically is composed of these layers of polysaccharide moieties, and these are interconnected. These are interconnected, like your vulcanization of rubber. You know, vulcanization of rubber means the rubber layers, the isoprene, polyisoprene units are having disulfide, trisulfide, all these bonds, and that makes the whole thing very rigid, and that made the possible for the development of tires. It's the same principle bacteria utilizes. So this is the sugar moiety, and the sugar moiety has hanging pendant groups which are actually made up of peptides, means amino acids, and then there is a small peptide hanging on this right side and a small peptide hanging on the left side, and then they are joined together. So that makes a cross link. So what happens ultimately, the whole thing becomes cross link. As I said, that the whole thing becomes cross link. There are a lot of cross links like this, and the wall becomes very rigid. Okay, so that is the biology behind this cell wall. So basically, again, I repeat that without this cell wall, the bacteria cannot survive because the internal osmotic pressure is very high. And what is then the cell wall is made up of? The cell wall is made up of what is called peptidoglycan, because these are sugar units which are called glycan units, also called glycan units. So glycan units, and they have peptide units. So they are called peptidoglycan. And peptidoglycan biosynthesis means this cross-linking between these hands. That is what is stopped by the penicillin. That is what is being stopped by penicillin. Okay. So in short, penicillin basically interacts with the bacterial cell wall biosynthesis. It's very good because the human does not have the cell wall. The cells are not covered by walls. It is covered by membranes, lipid membranes. So that's why penicillin is so specific onto bacterial cells. Now, as chemists, we must be very curious that what are these amino acids? What is this cross-linking reaction? So let me show you the cross-linking reaction. Actually, forget about this. This is a sugar backbone, and this is the peptide pendant groups. The pendant groups have only hands like this pentaglycine. This glycine has a NH2 at this end, and this peptide, which is the pendant group, that has got two amino acids at the end that is made up of D-alanine and D-alanine. And what happens here, the chemistry that goes on is that this NH2 breaks the peptide bond between the D-alanine and D-alanine, and these goes out. So what happens, that basically this is suppose the glycine, and this is your D-alanine and another D-alanine. This is the peptide bond. Remember, this is very unique. These are D configuration. And this glycine NH2, with the help of an enzyme, again I repeat, this is not spontaneous. This is an enzymatic process. So it needs a catalyst. Okay. And that hydrolyzes, that removes this alanine, and alanine goes off, and this alanine is now linked to the glycine moiety. So this is what is nothing but called transpeptidation, like transesterification. One alcohol moiety is replaced by another alcohol moiety. So this will be called transpeptidation. So one amino acid is replaced by another amino acid. Here, D-alanine is replaced by glycine. But in the process, it becomes cross-linked, that there is a linkage between these two pendant groups. So this happened in all these sugar backbones, and ultimately the bacterial cell wall becomes very rigid. Okay. And I think I skipped this slide. I will write now exactly what happens. See, if you write D-alanine and D-alanine, you can say that why does penicillin stop the action of that very enzyme, which does the cross-linking, Is it okay if I writing is clear? Huh? Which does?
Yes, absolutely clear.
Which does the cross-linking. So that is the question now. The big question is, how does penicillin stop this cross-linking process? Remember, this cross-linking process is carried out by a catalyst, which is an enzyme, and the reaction that takes place is a transpeptidation. So what you call this enzyme? You will call this enzyme transpeptidase. Okay, that is, that is how the enzymes are names. If something causes hydrolysis, that is called hydrolase. Transpeptidase is causing transpeptidation. Okay. So the question is why penicillin stops this, or how it stops the action. Remember, if you want to go to the resistance later on, we need this chemistry to be unraveled. Okay. So there was a person at Harvard University, Harvard Medical School, basically. His name was Tominger. He actually gave a hypothesis, and that is still the valid hypothesis, what he said. He said that if you write D-alanine and D-alanine, it will look like this. This is one of the conformation. We are, as organic chemists, we know conformation very well. Okay. So this is D-alanine, D-alanine. That is, remember the last two amino acids in the sugar backbone. The last two amino acids are D-alanine and D-alanine. And this is one of the conformation of D-alanine, D-alanine. Okay, this, if you work out the configuration, whether it is L or D, you can work out. You will see that this is actually D and this is also D. Okay. And if you write the structure of penicillin, this is what is the structure of penicillin. You will see that, interestingly, a major portion of penicillin mimics the structure of D-alanine, D-alanine. A major portion of penicillin mimics the structure of D-alanine. Like this, you have the carbonyl, you have the NH, you have this backbone, you have this CO2H, you have this carbon. You look at penicillin now, this part, that part, this part, the carbonyl, this part, and that part. Okay. This part also can resemble the CH bond. So there is a great resemblance, and these hydrogens may be mimicking the methyls, maybe. Okay. That was his hypothesis, that a major portion of penicillin resembles the D-alanine, D-alanine in its one particular conformation. And not only the conformation, that absolute configuration is also matching. So what happens, the enzyme, instead of attacking the D-alanine, D-alanine, see, the transpeptidation reaction is nothing but that a serine hydroxy group, serine is another amino acid, which attacks the carbonyl and hydrolyzes the peptide bond. Okay, this is the enzyme, transpeptidase enzyme. And then the transpeptidase gets attached to this part of the peptide chain, and then water, basically water comes and water hydrolyzes the enzyme. I can make it a little bit simpler by writing the mechanism. See, the mechanism is nothing but, and again I write D-alanine, D-alanine, hydrogen, 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 and this is the NH, there is this methyl, and there is this hydrogen. So the mechanism is like this, that the enzyme has a amino acid, which is called serine, which has got weight. This weight attracts the carbonyl. You know that first the carbonyl, the electron flows like this, it goes to the oxygen, and then it comes back and breaks this peptide bond. So you get what is called an acyl enzyme. This is the acyl group. The enzyme is now attached to this part of the peptide chain, and this is the enzyme. And now water comes. See, the other D-alanine has come out. Other D-alanine goes out. Water comes and frees the enzyme, so that it is basically a catalyst. It is involved in the first step and it is released in the second step, and then it can again go back and do the same reaction. Okay. Now, if you write penicillin, as I said, the enzyme is fooled by the structure of penicillin. It is just fooled by the structure. It thinks that this is my substrate, the enzyme thinks. So it attacks the same way. Carbonyl, it goes there, comes back, and this goes away. What happens now? This enzyme, which is attached to this molecule now, NH, CO2H, this is now stable. Okay. And what was the second step? Enzyme has to be freed. This step is extremely very slow. So what happens? This enzyme, which is absolute necessity for the synthesis of the bacterial cell wall, this is stuck to the penicillin. Okay. And thus the enzyme cannot work. See, if I hold you are writing something with your right hand. If I hold your right hand for a long time by an external agent, you cannot write. See, your handwriting is inhibited. The same thing is there. Penicillin is mistakenly regarded as the substrate by the transpeptidase enzyme, attracts this. This is called a beta-lactam ring, which is extremely reactive. You know, small membered rings are extremely reactive, and this is taking place, enzyme attacks by the same OH group, but it is now it is very stable. It is not hydrolyzed by water, unlike this. This is very fast and this is very slow. So what happened? This will be accumulated in the bacteria, and in the process, bacteria cannot make the cell wall, and the bacteria ultimately dies. This is the mechanism of penicillin, and in fact, this is the mechanism of many antibiotics that we see today. That is starting from, I think I have a slide. Let me just check very quickly. Yes. Cell wall synthesis disruptor, I call them. Cycloserine, vancomycin, bacitracin, penicillin, cephalosporins, monobactams, carbapenems. So a major portion of our antibiotic that we have, the antibiotic arsenal that we have, major portion attacks the cell wall. There are other ones like ciprofloxacin. You have heard of cifran. You have heard of streptomycin. You have heard of many other antibiotics. But a major portion, again I repeat, they attack the cell wall because they are extremely selective. Host does not have cell wall, but we have DNA. So if anything attacks DNA, so that will definitely be attacking some of my DNA also. So they are more toxic. Other antibiotics are more toxic than the cell wall synthesis disruptor. That's why penicillin is so harmless in our body. Okay, so let me go to the next slide. This was what was going on, that the mechanism was established, that the bacterial cell wall, how the biosynthesis was stopped. People also got the crystal structure of the transpeptidase. Everything was known. But bacteria is also very clever, like the virus, what we see in this pandemic. They keep on changing, but bacterial mutation is much slower. Like virus mutation is very fast. Bacterial mutation is a slow process. So what happened during the course of this antibiotic use, bacteria developed a mechanism that if you add penicillin to that type of bacteria which has become resistant, what is the mechanism of resistance? See, I said that initially the transpeptidase will hydrolyze this beta-lactam ring and it stays there for a long time because it is not hydrolyzed. But bacteria over the years came out with another set of enzyme which is called beta-lactamase, Again, from the name I can realize lactamase means something which breaks the lactam ring. So lactam ring is which one? This is what is called the lactam ring. So this lactam ring is broken by this new enzyme. This is not transpeptidase, remember. This is beta-lactamase. And what is the difference with transpeptidase? That beta-lactamase have an ability to immediately bring in water and release the enzyme. It is extremely fast. So what happens? The penicillin you are giving, the beta-lactamase immediately, ultimately hydrolyzes into this CO2H and NH, and there's this 5-membered ring. Okay, I'm not writing the full structure. This is harmless. This is not an antibiotic. So this beta-lactamase enzyme now is making this penicillin ineffective. How? Only difference with transpeptidase is that it can make the second state, which is hydrolysis, very fast, extremely fast. It's a turnover. You know, enzyme has a something which is called turnover number. That means a catalyst has a turnover number. That means each molecule of the catalyst can catalyze the reactions of how many molecules. This beta-lactamase enzyme can have a turnover number of 10 to the power 6. So one beta-lactamase molecule can hydrolyze 10 to the power 6 molecules per minute. So there is a unit of time. Okay. So it's a very efficient enzyme, but it actually immediately heralded a signal which is very bleak for the human survival, because all the penicillins that we were having at that time, they were being making ineffective by this beta-lactamase. So what we have learned so far, we have learned that there are antibacterial agents, and between the in one of the class of antibacterial agents is antibiotic. The first antibiotic to be discovered was penicillin, and then penicillin. What is the mechanism of action of penicillin? It resembles D-alanine, which is a very important component of the bacterial cell wall, and then the transpeptidase enzyme, which does the cross-linking, that is inhibited by this penicillin. Okay, how? The transpeptidase attacks the penicillin molecule because it has a very strange beta-lactam ring, so it can attack the beta-lactam ring, and then the enzyme gets stuck to that molecule. Water does not come and hydrolyze and freeze the enzyme. The enzyme cannot be freed to catalyze the next set of reactions. So enzyme gets stuck to the molecule. That is the mechanism of penicillin. But then the bacteria, over the years, thinking what to do, because in this world, Darwinian theory, survival of the fittest, everything has the right to live. So bacteria started thinking how to fool the mankind to develop some arsenal to kill the penicillin. So what they did, they actually have this beta-lactamase. They developed this beta-lactamase enzyme. What is the difference between the two? The beta-lactamase in transpeptidase, hydrolysis does not take place. In beta-lactamase, hydrolysis is super fast, high turnover number. So penicillin was being made ineffective. Okay. Because of time shortage, Hoymanti, you will have to tell me when I have to stop. Huh? So the next is that what to do. We were very lucky that in 1980, there was a molecule which was called clavulanic acid. This is the structure. Yeah, Hoymanti, you are there?
Yes, sir.
Yeah,
You
how
can
much
carry
time
on.
I have?
You can carry on, no problem.
Okay, no problem. Okay. So this clavulanic acid, this was also isolated from a microorganism, because there was a fanatic race between the different pharmaceuticals to come out with a molecule which can overcome this resistance problem. Resistance means this beta-lactamase enzyme. You have to now use a molecule which stops the transpeptidase enzyme from working, and which also stops the beta-lactamase from working. Then only you can overcome this bacterial resistance. Okay. So people were actually trying to, one of the ways is to isolate natural products. Isolate natural products from where? See, Louis Pasteur gave a theory that if you have a mixture of bacteria growing in a culture, then what happens? There is a fight between the bacteria. Like we are seeing fighting today also, Ukraine, Russia. There is always fighting. Who wants to survive in this world? So bacteria also survives against, fights against each other. Okay. And how do they fight? Their fight is basically making chemicals to kill the other bacteria. And that is the reason actually bacteria, some of this fungus or some of the bacteria makes penicillin. They don't make penicillin for us. They make penicillin for themselves to survive, because they make penicillin, put it on the outside, and then that kills the other bacteria. Okay, so that is how they do. So basically what people were doing at that time, pharmaceutical companies isolated the bacteria which have got resistance and trying to force them with other bacteria so that to kill them, and so that they can come out with new chemicals. Okay, and so that was being screened. So different bacteria which are present in, these are mainly present in soil bacteria. They are actually trying to get compounds from the soil bacteria, and suddenly there is a compound, there is a company, now it is called, the company name has changed. At that time it was called Beecham. Okay, Smith, at one point it was Smith, Klein and Beecham. So they isolated a compound which was called clavulanic acid. If you look at the structure, very similar to penicillin, but some differences are there. One is this oxygen in place of sulfur. One is this double bond. And this double bond has actually made our life, so actually they rescued us from the onslaught of the beta-lactamase. What happened? Beecham scientists found that this molecule is not a very good antibiotic. That means it is not recognized that well by transpeptidase, but it is a very good inhibitor of your beta-lactamase. Inhibitor means stops beta-lactamase from working. So if it is a good beta-lactamase inhibitor, then what you can do? You can add this clavulanic acid with a penicillin like amoxicillin, and this combination you can give to the patient who is suffering from an infection caused by resistant bacteria. So what will happen? Clavulanic acid will kill the beta-lactamase or stop the beta-lactamase, and amoxicillin will stop the transpeptidase. This is a life-saving drug. I don't know. You must have heard the name of this drug called Augmentin Duo. Duo means basically, basically you have two compounds, amoxicillin, you have clavulanic acid. So now it's a combination. So when you have two enzymes to target, you can have this combination therapy. You know, HIV also is like that. HIV infection, that AIDS, you have combination of drugs. One goes for one enzyme, another goes for another enzyme. Okay. So this clavulanic acid, the question is how it is an inhibitor of beta-lactamase. It is an inhibitor of beta-lactamase because the first step is same. This hydrolyzes the beta-lactam ring. Okay. But because of the presence of this oxygen and the double bond, this is very important. This is nothing but an enol type of compound, an enol ether. This is an enol ether, which is very susceptible to opening. So immediately the nitrogen lone pair flies, and this opens up and becomes a carbonyl, and this becomes the double bond picks up the hydrogen. So this is your, so what happens now? So this is the story. This is actually, they are writing it in the enol form. It can go to the keto form also. And then, so you are basically having a double bond now here. You are basically putting a double bond here. But because there is a carbonyl, you know that alpha beta carbonyl is much more stable than beta gamma carbonyl system. So there is an isomerization that takes place. The double bond shifts from here to there, and that becomes a good Michael acceptor. Alpha beta unsaturated carbonyl compounds are good Michael acceptors. Now see, this nucleophile, this is the enzyme. The enzyme has started working on the beta-lactam, hydrolyze the beta-lactam, it creates this molecule, and now water comes and frees the enzyme. That is the normal process. But because of this Michael acceptor, The enzyme is a big molecule. So it has got nucleophile also on other positions. That nucleophile now attacks at this double bond and undergo a conjugate addition, that is Michael addition. So the nucleophile is now attached to the molecule, and this nucleophile actually will be hydrolyzed. This can be free actually. This is something wrong here. This is free by water. Water can come and free this part, but the enzyme overall is now tied up to this molecule. You understood? Basically what happened? In short, I can say that you have a molecule A. The enzyme thinks that this is my substrate. So it works on the normal procedure, that means hydrolysis of this beta-lactam. So it forms a compound which is A, say, prime, suppose. Now this A prime undergoes some chemical changes and become F. Why I say F? Because F stands for a story, you know, Frankenstein. Frankenstein story. So F is becoming a monster. Why it's a monster? Because the enzyme gets stuck to this molecule. So the enzyme is now reacting with this F and form a covalent bond with this. And when you form a covalent bond, it is not easy to free the enzyme. So this is what is called a Frankenstein molecule. Actually, you know, Frankenstein is the scientist who developed who gave life to that, to the dead body. And generally we think Frankenstein is that monster, but actually Frankenstein was the scientist. Here, the real Frankenstein is that it's a scientist. Here, scientist is nothing but the beta-lactamase enzyme. But this is very like the Frankenstein story, that this molecule opens up and ultimately kills the very host molecule, which is the enzyme, and kills the enzyme. This is killing the enzyme because the enzyme is attached by a covalent bond. Okay, this is also called a suicide inhibition. Why suicide? Because the enzyme is basically virtually committing suicide by gulfing, by accepting this molecule. Okay, so this is the story. Now, this is how this clavulanic acid saved us. But the problem came later on, in the specifically in 2009. 2009, there was an infection that happened. A person came to New Delhi. He was sick in a hospital, went back to Sweden, and then he suffered from a bacterial infection. And that bacterial infection could not be cured. They tried all sorts of antibiotics and nothing worked. And he died. Okay. Then they isolated that bacteria, that what is this bacteria? And they found that this bacteria is little different from the beta-lactamase, which I told you earlier. Earlier, beta-lactamase uses a serine wedge to hydrolyze the beta-lactam ring. So that is called serine beta-lactamase. But this the new bacteria that was isolated in 2009, that uses a metal ion to hydrolyze the penicillin. A metal ion like zinc. Okay, so zinc is chelated to this oxygen, and another zinc ion, there are two zinc ions, that is chelated to the nitrogen. So now water comes, so you don't have any participation as such by the enzyme. Enzyme only brings these zinc ions, so these are metalloenzymes. So these metalloenzymes, they also hydrolyze penicillin, making it ineffective. And interestingly, clavulanic acid does not work against these metalloenzymes. So life became very complicated after 2009. And unfortunately, this bacteria, which originated from New Delhi, was called NDM. NDM harboring bacteria. What is NDM? New Delhi metallo-beta-lactamase harboring bacteria, which is kind of very bad. No? Our capital is associated with an infectious disease. So government protested to the WHO. Actually, the paper was published in Lancet. There is a journal called Lancet. Still, people are calling it New Delhi metallo-beta-lactamase. But there was some papers by Indian scientists. They started calling it PCM, plasmid carbapenemase metallo-beta-lactamase. We will not go to this controversy. It is still generally known as, we will call it as metallo-beta-lactamase. And if you type in the internet, you will see NDM. There was no cure, almost no cure for this NDM harboring bacteria. In between, okay, some molecules were the last line of defense. What is last line of defense? That you start with a weaker antibiotic, and then you slowly, if that does not work, you go to stronger antibiotics. So what happens? The stronger antibiotics, they were also failing. And the problem in our country is that generally the antibiotics are so misused that weaker antibiotics may not work. So the doctor prescribes the stronger antibiotic or the strongest antibiotic to start with. The problem is, like if you are in a war with someone, and if you show your strongest weapon to the enemy from the very beginning, you are becoming weak. You have to slowly divulge your arsenal, your whatever you have, rifle, then your missile, ultimately you have atom bomb. So similarly, we should not use the atom bomb. We should not use the strongest antibiotic like that. If you use the strongest antibiotic, what happens? The bacteria now knows the character of the antibiotics, because always think that we cannot really kill all the bacteria in our body, because it is just a half-life type of cases that 100 will go to 50, 50 will 25, 25 to 12 point. Some bacterial colonies will remain. So they will remember that what antibiotic has been used last time. So if you have used your last resort of antibiotic, then when these bacteria becomes resistant, that last antibiotic will not work. And that is what is the scenario today. Our last resort almost gone. There is only one antibiotic that is called polymyxin, but which is very toxic. That is the last resort. So many people today are dying out of pneumonia, COVID cases, because of secondary infections of the lungs due to these resistant infections, because no antibiotics are working for these lung infections. Okay, because we have already shown our last resort to the bacteria. So bacteria has come out with this metallo-beta-lactamase, the New Delhi, the NDM ones. Okay, so what to do now is how to come around with this problem, around this problem. One is finding suitable inhibitor of MBL. Another is that the doctors also should be very careful. They should really first, when somebody goes with a fever, they should not use the strongest antibiotics to start with. They should first know that whether this bacterial infection has NDM or not. If you use polymyxin, then what will happen later on? Polymyxin will become resistant. So there must be a very quick way to really know that what type of bacterial infection the person is suffering. The problem is to know that it takes about 72 hours. The culture, the culture reports come after 72 hours. So you have to have rapid diagnostic assays to know what is whether there is NDM present or not. The other is that is not a solution. The other is that will actually check the spread of the bacteria. The other is that finding suitable inhibitors of this metallo-beta-lactamase. And some people are trying to make chelator of zinc to stop this metallo-beta-lactamase from working. Our group in our laboratory, what we are trying to do is basically how to quickly determine that whether an infection is due to NDM related bacteria or not. It is a huge task. It's like catching a particular fish in a galaxy of fishes. You want hills of fish to be caught, but that is not possible. There are so many fishes. But if you have a fish hook, which is very specific for a particular kind of fish, you can take out the fish as far as your wish. So, but the fish hook has to be very specific. So similarly, see, everything has a chemical counterpart to it. If you have different proteins, what we are talking about, that a bacteria containing different proteins, like NDM, it can have NDM, it can have transpeptidase, it can have all these RNA polymerase, DNA polymerase, everything is there. You have to identify whether your NDM is present or not. How to do that? This is a cartoon picture. You have 4 proteins. 4 proteins, proteins have receptor sites or active sites, and that depends on the, that has a particular geometry. So what you do, you make a compound where there are 3 functionalities, okay? And you add to this mixture of proteins. One functionality is such that it recognizes only one of the cavity or the active site of the protein. In this case, C. So you see this hand has bound to this enzyme, which is C, okay? And when it is bound, others are free because this hand is not binding. These are not complementary. So then you do some chemical reaction, usually done by light. So you shine light, and there is some group which forms a cross-link, which forms a bond with this part. This is called a cross-linking agent. And this is an agent which gives fluorescence. So now what you do what is called a gel electrophoresis, because proteins are charged molecules, so you can move them under a matrix. So they will move under the matrix, 4 proteins. These 4 proteins can give, you have different coloring agents, okay? So there is a coloring agent which can color all the proteins, blue color. But amongst one of these 4 proteins, one is cross-linked. And that cross-linking can give fluorescence. So before you stain this with the blue, which is called Kumasi blue, you see it under the fluorescence microscope. And you see a band in the gel, okay? You take a picture of that, and then you same gel, you see under the Kumasi blue. And now you compare. So this is your enzyme C, you see where it is cross-linked. So this is the way. If you have many enzymes, that is also possible. Important thing that this should be very specific, that your hand should be very specific. So the design of this is very important. And ultimately, so I think the time is almost over, right, Hoymanti? So students,
No, sir.
I don't want to go.
You can carry on.
No, I will just 5 minutes, I will wind up. Okay. So this is the experiment that is done, the gel electrophoresis. This is the matrix. You add the protein mixture, you take the gel out, first see it under UV, and then you stain. Okay. This I will say, this is the ultimate picture that you see in the blue Kumasi gel. If there are too many, you see too many proteins here, right? But if you have this, what is called protein capture agent, that tripodal molecule, okay? If you again go for the design, what I said, the design should have 3 hands. One is called the affinity hand, means which will bind to the enzyme. Another is called cross-linking hand. That means a functionality by shining light, which can form a cross-link. And another is called a visualization hand. In our case, it is fluorescence hand. So this is the template on which you attach these 3 hands. Okay. So like here, I show you, this is the template. There is this sulfonamide. Sulfonamides are good inhibitors for many enzymes. Azide, azide can shine light. It will form nitrine, and that can form cross-link. And this is a pyrene moiety, which can go into fluorescence. Okay. So these are the 3 hands. And we this was our initial molecule, but then we thought that this molecule is too big, because after all, enzyme has to accept this molecule. If the cavity is very narrow, then you need a small molecule. So ultimately what we did, See, this is the picture, actual picture. We had these 3 enzymes and this is under UV. So the molecule works, but it is not a very useful molecule in the sense that it is too big. So what we did, we changed the molecule and we changed it into the template is itself fluorescent and it itself has a photo cross-linking hand, and we just add the selectivity or the affinity hand to it. Okay, so I will show you the last few slides. So we made this molecule. This is much smaller. This is the fluorescent hand with an azide already there, and this is the affinity hand. Okay, this is the affinity hand. Sulfonamides, as I told you, they can bind to, maybe I have not told you, they can bind to zinc ions very easily because they are very good chelating groups. Okay, so first we saw that whether this molecule works onto metalloenzymes or not. There is a metalloenzyme which is human carbonic anhydrase. This is a zinc-based enzyme. So this is the same experiment. This is under UV and this is under Kumasi blue, that blue stain, and you see that yes, HCl2 can be seen selectively. So we repeated the experiment. This is the NDM. All these slides are there. This is the slide how enzyme hydrolyzes. So we made this molecule, different molecules, which you will see. This is the hand, fluorescent hand, along with the cross-linking hand, and it has got different functionalities here, which are chelators of zinc. These are chelators of zinc. And ultimately what we established that this is the NDM band, and this NDM band can be seen with our model compound. This is the model compound. This is another model compound. This is the one which works the best. That if you have many enzymes like this is actually clinical isolate. You isolate the bacteria strain from a person who is having NDM. This is the NDM. So this is the blue stain, and if you look at the corresponding lane here, you see the NDM. So that means this roughly takes about 2 hours, the whole experiment. So within 2 hours, you can detect the presence of NDM in the bacterial strain that is present in a human who is suffering from an infectious disease. So that's a much quicker way to know the existence of NDM. Actually, this technique can be applied to detect many enzymes. What you have to do, you have to just change the affinity hand. So this is the story I wanted to tell you. So just in short, what I have said, that I told you about the antibiotics, their resistance mechanism, their mechanism of action, then beta-lactamases, serine beta-lactamase, clavulanic acid works, but now new strain has come, NDM, for which it has not worked. But so the urgency of the day is that how to find an inhibitor of this NDM and how to detect it very fast, whether it is present or not, so that we unnecessarily don't use your colistine or polymyxin. Okay, so we have developed this technique which is called affinity guided protein labeling technique. Okay, so that is the summary. So I think that actually this is the slide which actually explains everything, that you are catching. The proteins are actually basically representing different fishes. So you have fish hooks for different fishes, and you can catch your particular fish of your liking, pomfret or whatever, INSA, whatever. In our case, it is protein. You are chemist, so you want to isolate the protein or detect the protein, and that is it. So by this technique, you can detect the NDM very rapidly. So these are some of my workers, my co-workers. This is also, there are a lot of biology. These are my students. Okay, she's, I think, also doctor, and Professor A.S. Ghosh, Biotechnology Department, IIT Kharagpur. Professor Amit Das, Dr. Shantini Roy was a postdoc. Chris Cofield, he is actually the chairman of the organic chemistry at University of Oxford. I constantly have touch with him, and he is working on development of inhibitors of NDM. And I was working on how to detect NDM very rapidly. So that's it. These are some of the funding agencies. And this is the just a last slide to the students specifically, that never think that life is very straight. Life is like this, and you really don't know what is there at the end. So you walk down in a zigzag fashion, obstacles will come, but ultimately you come here and you see a new challenge and you continue. Okay. This is kind of a slide, philosophical slide, that at one day sun has to set. So at one day I have to quit the research. But I still try to do collaborative research, especially this NDM related bacterial resistance. Thank you very much.
Thank you, Professor Basak. Thank you so very much for this extremely simple, lucid, storytelling type of a webinar. I think that the students have thoroughly enjoyed it. The house is now open for questions. Students, please ask your questions one after another to sir. Please unmute yourself and ask.
Hello, can you hear me?
Yes.
Yes.
Sir, I Dr. Shankar Prasad from Behala College.
Yes.
I feel proud to be a student of your Guru, FASc Tolapatra. FASc Tolapatra to in a class. But Amit Basak is my best talented student. So Amit
But
Basak
don't
is
shoot.
grand name in the world. We
No.
are proud for innovating, illuminating, and encouraging lecture, which will help to the not only to the students. This lecture will also give immense. It will also immensely help to the teachers like me. Now I like to share some questions or some
Yes,
problems
please.
with you,
Yes.
sir. Which penicillin was discovered by Fleming? Because penicillin is a group of compounds we know. So which penicillin was discovered by Fleming?
Which penicillin was discovered? Fleming actually could not isolate the compound. In the actually, when these microorganisms make penicillin, they what they do, they make the first compound that they make is all the compounds. Remember, this ampicillin, amoxicillin, all are semi-synthetic penicillin. So the compound that is present in the fermentation medium was a side chain at this position, which is alpha amino adipic acid. Alpha
Sir,
amino
is it
adipic
probably
acid.
penicillin G or penicillin
This
like
is
that?
not a good antibiotic. So
No.
can you see the slide? You
Yeah,
cannot
yeah,
see
yeah.
the slide. Huh?
No problem. I have
Can you see
heard
the
your
slide
language.
now?
Okay, no problem.
Yeah. What happened actually? He could not isolate or characterize the structure, but actually, when this bacteria or fungi make the penicillin, they make it in the form where the side chain is alpha amino adipic acid, and then it was realized that we don't need this alpha amino adipic acid side chain. What we need is benzyl side chain or phenoxymethyl methoxybenzyl, like penicillin G, penicillin V, etc. So then there was a method that was available, fermentation medium, a way that you can get NH2, not the side chain.
Yeah.
So you get NH2. So NH2.
Okay,
Now,
okay.
if you get the NH2, you can put, this is a simple chemistry. You do acylation with any acyl fluoride.
Different types of penicillin can be derived after acylating group, changing R group. Okay.
So that's what happened. So Fleming,
Okay.
Fleming, I believe he was handling this alpha amino adipoyl penicillin. Sorry.
Sir, I like to know from you, sir, penicillin resistance or antibiotic resistance is a problem to us. But another thing, persistence of antibiotic in our body, that can cause what type of problems?
You know, although these antibiotics, some of the antibiotics, I say penicillin is harmless. But there is one problem. The problem is that we generally suffer from, there are two kinds. One is some people have sulfur allergy.
Yeah.
If they have allergy to sulfur, then penicillin has sulfur. Even sulfonamides have sulfur. So that type of antibiotics cannot be given. If you use it for too long, then what happens in our intestinal gut? There are many helpful bacteria that are present, which are making the B complex group of enzymes, a B complex group of cofactors, the coenzymes, not the enzymes, the coenzymes. Many of these vitamins are made by these bacteria. So those bacteria will be killed. And so you will get indigestion. You will suffer from diarrhea. So a major problem of the antibiotic like penicillin, any antibiotic, will be that you will be suffering from some kind of stomach upset. That is because of this killing of the helpful bacteria that are present in our intestinal gut. So you have to take what they say, that you have to take a lot of GARD, or you have to take lactobacillus tablets available in the market, lactobacillus, which are helpful, which can make some of these vitamins, which help in our digestion process.
Sir, antibiotic resistance can be permanent for a person.
Permanent means actually he will be killed, means he cannot survive. If you cannot, ultimately, if you cannot kill the bacteria, so bacteria will continue to grow.
Suppose once upon a time it was resistant, but after some time it can be it cannot be resistant. It may happen sometime.
It cannot be resistant sometimes. Means a resistant going to non-resistant. That's what you are saying. That can happen. There is constant mutation. Some mutations may be helpful, like our COVID. It is changing. Some mutations may not be, actually may be good, that it is not binding to the spike protein has changed, it is not binding. But some mutations may be very bad. So it is actually a chance also. So that is possible, non-resistant bacteria going into non-resistant. That depends on the use of these antibiotics. The more antibiotics is used, like a lot of people are anti-vaccine, We are hearing that in USA, a lot of people are against taking vaccines against the COVID. Okay, there is a risk. The more you take vaccine, the bacteria will mutate, the virus will mutate more. The more you use the antibiotic, the bacteria will mutate more. Okay, so there is a chance of always the resistance.
Okay. Sir, I have one query. Sir, antibiotics are used usually for infection caused by bacteria, and antiviral drugs are used for viral infection. But is there any drug which can be used both for antibiotic and for antiviral?
No, I think that is actually an entirely different mechanism. Viruses replication and amplification is a completely different chemistry than the bacteria. Our problem is many of the fever that we have, we go to the doctor and we ask the doctor to prescribe antibiotics. Actually, we will not need any antibiotics if it is a viral infection. It will go, some people say that it will go in 1 week, some people say that no, it takes 7 days to be. So basically, in many of the viral cases, we are unnecessarily taking antibiotics. And that is one reason that why bacteria is becoming resistant. You understood what I said? Actually, the mechanism of their life cycle are totally different. So it will be very difficult to have a compound which can kill both.
Sir, some recent report informed that the sulfonamide drugs are antibiotics, but sulfonamide drugs can also be used as a HIV treatment.
No, so you see that it can be changed. You are changing it can be sulfonamide, but you have to change the skeleton. Chemically, you have to do some chemical modification. You have to do. Okay, so that will be possible. I will tell you one story. Like there is a drug which is called, which was given to the, which caused birth defect. I think I am forgetting the name. The story, which was given as a racemic mixture, and then there were a lot of babies born with birth defects, and they found that the compound which is causing birth defect later on that is being used as an anti-gout or anti-rheumatic agents. So, so they might have other
thalidomide drugs.
See, this is what is called
Thalidomide drugs.
Huh?
Huh?
Thalidomide, right?
Thalidomide.
Thalidomide, correct. Thalidomide.
Okay, sir.
So the bad one is now used.
Yeah. Two isomers are there. One isomer, an isomer can act for birth defect, and another isomer is used for other purpose.
Yeah,
Okay,
but the
thanks a
bad
lot.
isomer is now used for other purposes. Okay.
Thanks a
Okay.
lot, sir. Thanks a lot.
Thank you. There is a question from our student, Trijita Roy. She has asked that among all the antibiotics you discussed, sir, which one is the most effective with respect to antibiotic resistance?
Okay.
Which
Today,
drug-
very good question.
And there is
Which
another
drug?
part, and
Okay.
he has asked which drug acts most effectively to prevent antibiotic resistance?
Today, actually, we have, I can show you the structure. I actually had 3 structures here. Just a second. Very good question. What happened, actually, people were making different antibiotics, different types of antibiotics, randomly or also rationally. Okay, now this is the structure of penicillin. So what you can do, one, as chemists, we can do this, as I told, the acylation. You put different R groups that give us amoxicillin, ampicillin, cloxacillin, all these things. Okay. But the problem is the main skeleton, which is the pharmacophore. This is what is called the pharmacophore. That is causing the activity. You are not, that remains the same. So whatever, see, like whatever ornaments you wear, you remain the same person. People will recognize you very easily. So these are nothing but ornaments, R, changing the R. So if you want to tackle this problem, you have to change the skeleton. Skeleton means the pharmacophore. So with this idea, what happened? Let me see. There is a compound called meropenem. Where is, I used to have the structure. Yes. This is the compound, meropenem. Then there is now another compound, ferronem, which is being prescribed today. These are the compounds which are effective against also resistant bacteria. But if a person is having lot of infections, already septicemia has started, and if he or she has very weak immune system, okay, then they may fail. But these are currently the best antibiotic. These are called carbapenem antibiotics. Meropenem, ferropenem. What is the other question? So
she asked that which drug acts most effectively.
Yeah, so this is the drug which is prescribed now in extreme conditions. Okay.
Okay.
And
Srijita, are you satisfied, or you want to ask some more questions?
No, ma'am. Till now it's okay.
You can now search in the internet. You see this trimeropenem, okay, or carbapenem.
She's a semester 6 student. Another question is there from Muslima Nazneen, semester 4. She has asked that gram-positive bacteria have a peptidoglycan layer on the outside of the cell wall. Gram-negative bacteria have peptidoglycan between membranes. Will penicillin work better on gram-positive bacteria?
Yes, I think that's a very good question. Actually, this is you said that this is the cell wall, this is the outer membrane. Gram-negative bacteria, you said gram-negative bacteria has a outer membrane layer and the cell wall is inside. So your answer, the first answer is that for gram-positive, it works better, much better, because gram-positive, the wall is outside. Okay. So penicillin works with gram-positive bacteria much better. And the resistance that I'm talking about, you have raised this question. I'm very happy. Gram-negative bacteria, it's difficult. And the resistance, this Indian bacteria, what we are talking about, they are all gram-negative bacteria. They are much more difficult to kill by the penicillin because of this reason, because of this membrane, which has got a hydrophobic barrier. So entering is a problem, but you have channels from where the molecule can go. And this is the cell wall. Okay. So to reach cell wall, before that, beta-lactamase is there, hovering around this. surveillance, like it's a battlefield. They are just constantly watching who is coming. So penicillin comes, beta-lactamase immediately kills the penicillin. Okay, so negative, gram-negative ones are the ones which are very difficult with the penicillins. And in fact, now, of course, meropenem can actually inhibit the beta-lactamase enzymes much efficiently. So, so to answer is gram-positive. It works with gram-positive much more effectively than gram-negative.
Okay,
And this is-
Muslima?
Yes, ma'am. Yes, ma'am.
Okay. Acha, Shreya Ghosh has raised a hand. Do you have a question, Shreya?
Yes, ma'am. Good evening,
Please
sir.
ask. Please ask, sir.
স্যার বলছি
Yes.
যে কি অনেক ব্যাকটেরিয়া তো রয়েছে যেগুলো কালচার করা যায় না, তাহলে সেই ব্যাকটেরিয়ার ক্ষেত্রে আমরা টেস্টগুলো কিভাবে করছি, মানে ইজ দেয়ার এনি কালচারাল ইন্ডিপেন্ডেন্ট স্টাডি?
অনেক ব্যাকটেরিয়া আছে যেগুলো যে সেটা হচ্ছে মেটাজেনোমিক্স, মানে মেটা there are so many bacteria which cannot be cultured, right? That is true. Those are extremely difficult. Unless you have what you can do, you can do the whole ensemble. See, all bacteria together. Like people, as I said, there are many bacterial colonies which are not being recognized. They cannot be grown also, as you said, which cannot be cultured. If they cannot be cultured, then you have, it's very difficult to really to do the conventional assay that I showed you, Petri dish. So it's very difficult. It's still a challenging problem. Fortunately, those type of bacteria are not invading our body yet, also, or we don't know. Okay, I think you got the answer, na? These are called metagenomics. Metagenomics is another area where you cannot really culture the bacteria. So you have to find the bacterial colony through techniques which are other than the conventional Petri dish, through other microbial techniques.
Is it
Like
okay?
if you go down, if you go down through the earth, okay, and you will see many bacterial colonies there which are not, that grows only under that condition. So it will be very difficult to grow under the natural condition. So you cannot grow them, okay. But a lot of research is going on in this area, that because you may get many, the other way around is that you may get very good chemicals out of that, which are like marine natural products. Why people are going into the marine? Because those are unexplored. Why people are looking for bacteria which cannot be cultured? But they will come when you will have technique to culture them. Like nerves, neurons cannot be cultured earlier, but now you can also grow the nerve cells. There are techniques. Hybridoma techniques, have you heard of? Hybridoma technology, that you add like 2 pieces of ice when you press and then release the pressure. That is called regulation. Okay. So if you have a bacteria which cannot be cultured, you press the bacteria with another bacteria which is very virulent, another cell, like cancer cell, which is extremely virulent, grows which is basically immortal. Press them, so you get a hybrid, and then you can culture them. So in that culture, you will get the characteristics of the other bacteria. It's like your Shukumar rice, abul tabul, that what was his combination of different species. It's the same thing. Hybridoma technology.
Shreya, have you got your answer?
হ্যাঁ ম্যাম স্যার আমার আরেকটা কোশ্চেন আছে।
বলো। আচ্ছা, আমি ইংরেজিতে বললাম, অসুবিধা হলো না তো ইংরেজিতে বলাতে?
না স্যার,
আচ্ছা বলো।
আমি কোশ্চেনটা ভালো বলতে বলে বাংলায় বলছি।
না নিশ্চয়ই
স্যার বলছি যে আপনি একটা স্লাইড দেখিয়েছিলেন যেখানে ডিফারেন্ট ক্লাসেস অফ অ্যান্টিমাইক্রোবিয়াল বেসড অন দেয়ার টার্গেট অফ অ্যাকশন। তা
রাইট,
ও টার্গেট অফ অ্যাকশন উপর ভিত্তি করে আমরা কি কোনোভাবে বলতে পারি যে এই টাইপ অফ ব্যাকটেরিয়া বেশি পরিমাণে রেজিস্ট্যান্ট হবে, এই মাইক্রোব্যাকটেরিয়া এজেন্টগুলো বেশি খুব রেজিস্ট্যান্ট হবে, এই টাইপ হবে কম হবে, প্রেডিক্ট করতে পারি কি?
না, ব্যাপারটা হচ্ছে এরকম, তুমি যত বেশি যে কম্পাউন্ড তুমি যত বেশি ইউজ করবে, তোমার রেজিস্ট্যান্সটা কিন্তু সেভাবে আসবে বেশিভাবে। এখন কেন সবাই, আমি তোমাকে এই তুমি যদি এখন ধরো ডিএনএ কে টার্গেট করো, ঠিক আছে, রেজিস্ট্যান্স ডেফিনেটলি আসবে। আজকে সিপ্রোফ্লক্সাসিন অনেক ব্যাকটেরিয়ার এগেনস্টে কাজ করে না। ওকে, so you have different brands, ciprofloxacin, then you have norfloxacin, then you have gatifloxacin. দেখবে আজকাল অনেক ফ্লক্সাসিন বেরিয়েছে, সব হচ্ছে ফার্স্ট জেনারেশন, সেকেন্ড জেনারেশন, থার্ড জেনারেশন, এগুলো কিন্তু সব রেজিস্ট্যান্স প্রবলেম। এখন এগুলো যত বেশি ইউজ হবে অ্যাট সাম পয়েন্ট অফ টাইম, আমার মনে হয় ওটা বোধহয় বলা শক্ত, ইট ইজ অ্যাকচুয়ালি ব্যাকটেরিয়ার পক্ষে মোস্ট ইজি হচ্ছে এনজাইম চেঞ্জ করা, এনজাইমটাকে চেঞ্জ করে দিচ্ছে, যেটা এখানে বা নতুন এনজাইম ডেভেলপ করছে বা এনজাইমের মিউটেশন করছে, সেটা ব্যাকটেরিয়ার পক্ষে খুব ইজি। তো আমি হয়তো বলা যেতে পারে যে যেগুলো ডিএনএ কে টার্গেট করে তারা হয়তো তাড়াতাড়ি রেজিস্ট্যান্ট হয়ে যাবে।
স্যার এনজাইমের ব্যাপারটা ঠিক বুঝতাম না, কেন মানে এনজাইমটা কেন বেশি ইজিয়ার?
মানে অ্যাকচুয়ালি তোমার ব্যাকটেরিয়াল এই যে অ্যাম্প্লিফিকেশন হোল থিং, অলদো ডিএনএ ইজ দ্য প্রিন্সিপাল অফ দি সেল, কিন্তু কাজ করছে কারা? কাজ তো করছে এনজাইমরা না, এনজাইমরা না থাকলে কিছুই হবে না। এখন তুমি বলতে পারো এনজাইম তো ডিএনএ থেকে আবার আসছে, ঠিক আছে, কিন্তু এই এনজাইমটা যদি কাজ করা বন্ধ করে তাহলে ব্যাকটেরিয়াল সেল অনেক ইজিলি তুমি, আচ্ছা তোমার চয়েস অনেক, কারণ অনেক ধরনের এনজাইম আছে, তুমি সেগুলোকে প্রত্যেকটাকে তুমি টার্গেট করতে পারো। ডিএনএ কে টার্গেট করতে গেলে তোমার লিমিটেশনস, কিন্তু সেই জন্য যত বেশি সেল ওয়াল সিনথেসিস ইনহিবিটর ইউজড হয় অ্যান্টিব্যাকটেরিয়াল এজেন্ট হিসেবে, তত বেশি কিন্তু বাকিরা ইউজড হয়। তুমি যদি পড়ে দেখো হাউ মেনি টাইমস ইউ হ্যাভ বিন গিভেন cephalosporin and penicillin and augmentin, সবাই এটাই দিচ্ছে, তুমি ডাক্তারের কাছে যাও এটা দেবে। অথবা যদি পেটের প্রবলেম হয়, যদি টাইফয়েড বা এই জাতীয় কিছু হয়, তখন এরা কাজ করে না, তখন কাজ করে মেইনলি হচ্ছে সেটা আবার খুব টক্সিক, সেটা হচ্ছে ক্লোরোমাইসিটিন। কিন্তু ক্লোরোমাইসিটিন আনফর্চুনেটলি খুব টক্সিক, বাচ্চাদের দেওয়া যায় না, ঠিক আছে, তখন ওরা সিপ্রোফ্লক্সাসিন দেয়, কিন্তু এটাও টক্সিক। আমি বললাম এগুলো হচ্ছে ব্যাপারটা হচ্ছে এখানে রেজিস্ট্যান্সের থেকেও বড় কথা হচ্ছে তোমার কি হয়, যত রেজিস্ট্যান্স হচ্ছে তোমার আস্তে আস্তে অ্যামাউন্ট অফ দি ড্রাগ তোমাকে বাড়াতে হচ্ছে টু ওভারকাম দ্য রেজিস্ট্যান্স। সেটা তো আরেকটা উপায়, তাই না? তুমি ঘুমের ওষুধ আজকে একজন খাচ্ছে, প্রথম শুরু করলো 0.25 ডোজ দিয়ে, কিছুদিন বাদে দেখে 0.25 কাজ করছে না, তখন 0.5 করলো, তারপরে ঘুম এলো। এটাও ঠিক তাই, তুমি ডিএনএ কে টার্গেট করছো, প্রথমে তুমি অল্প অ্যামাউন্ট দিয়ে দেখছো ভালো কাজ হচ্ছে, একটা ট্যাবলেট খেলেই হয় রোজ। আগে অ্যাজিথ্রোমাইসিন তাই ছিল, অ্যাজিথ্রোমাইসিন আমরা 3টে ছিল ডোজ, এখন 5টা থেকে 6টা থেকে বলছে 10টা খাও তবে কাজ করবে, ঠিক আছে। কিন্তু কি হচ্ছে, এরা যেহেতু এদের টক্সিসিটিটা বেশি, সেই জন্য প্রবলেমটা হচ্ছে তুমি ইনক্রিজ করতে পারবে না। তুমি এটা জানো তো যে একটা লিথাল ডোজ বলে একটা ব্যাপার আছে। লিথাল ডোজ হচ্ছে একটা ড্রাগ, তুমি যদি যেমন প্যারাসিটামল, সবাই বলে যে এত হার্মলেস, কিন্তু তুমি প্যারাসিটামল যদি বেশি পরিমাণে খেয়ে নাও তাহলে কিন্তু ইট উইল কজ আলটিমেটলি ডেথ। সেই জন্য প্রত্যেকটা ড্রাগের আছে, সেই জন্য প্রবলেম হচ্ছে এটার সুবিধা হচ্ছে যেটার প্রথম কথা রেঞ্জটা অনেক বেশি। তুমি দেখো কতগুলো ড্রাগ আছে এখানে, সেল ওয়াল সিনথেসিস ইনহিবিটর, ঠিক আছে, বাকিগুলোর কিন্তু অত, এখানে একটা আছে, কিন্তু এদের প্রবলেম আমি আবার বলছি, এরা যেহেতু প্রোটিন এই রাইবোজোমকে টার্গেট করছে, তাহলে এরা আমাদের রাইবোজোমকেও টার্গেট করবে। তা তুমি তো এদের ওই যেটাকে বলে যে উইন্ডোটা বলে থেরাপিউটিক ইন্ডেক্স, সেটা কিন্তু এদের অনেক বাজে দেন পেনিসিলিন বা যারা সেল ওয়ালকে যারা টার্গেট করে, ঠিক আছে।
ঠিক আছে স্যার, there are couple দুটো কোয়েশ্চেন আছে,
হ্যাঁ,
would you
বলো।
like to take them? Would you like to take them?
হ্যাঁ নিশ্চয়ই, আমার কোনো অসুবিধা নেই।
আচ্ছা, তাহলে একটা হচ্ছে কঙ্কনা মন্ডল, শ্রেয়া ছিল আমাদের পিজি সেমেস্টার 2 এর
হ্যাঁ,
স্টুডেন্ট।
ভেরি সবাই ভালো কোশ্চেন, খুব ভালো কোশ্চেন করছে, বলো।
কঙ্কনা is our UG Sem 6, ও জিজ্ঞেস
বাহ।
করেছি, is there a way to directly affect the AR gene to inhibit antibacterial resistance?
To the gene, see,
Is there a way
La-
to directly affect the AR gene to inhibit antibacterial resistance?
অ্যাকচুয়ালি দেখো প্রবলেম হচ্ছে যে আমরা যেটা করতে পারি তার মানে কি জেনেটিক ইঞ্জিনিয়ারিং করতে হবে, জেনেটিক ম্যানিপুলেশন করতে হবে, তাই না? জেনেটিক ম্যানিপুলেশন করতে হবে, সেটা কিন্তু অনেক ডিফিকাল্ট। হবে না তা আমি বলছি না, তুমি এমন করতে পারো, এমন কম্পাউন্ড ইউজ করতে পারো যে এই রেপ্লিকেশনটাই বন্ধ করে দিলে, এই রেপ্লিকেশনটা বন্ধ করলে, বুঝতে পারছি, এমন করলে যে একটা ডিএনএ এখানে ক্রস লিঙ্ক হয়ে যাচ্ছে, ঠিক আছে, একটা স্পেসিফিক কিছু ক্রস লিঙ্ক যেই হলো, তখন ডিএনএ স্ট্র্যান্ড তোমাদের মানে যেমন চুলের বেনি এখন যদি আমি আটকে দিই খুব জোর করে কিছু একটা দিয়ে, তাহলে তো খুলতে পারবে না, সেরকম স্ট্র্যান্ড তো খুলতে হবে ফর কপিং, ঠিক আছে। আমি যদি সেরকম রিজন বার করতে পারি তাহলে ঠিক আছে, কিন্তু এগুলো স্পেসিফিক বার করা খুব শক্ত, কারণ এটা আমার ডিএনএ কেউ করে দেবে, এই হচ্ছে ঝামেলা। কিন্তু দেয়ার ইজ এ ওয়ে, মানে আমি তোমাকে না বলবো না, দেয়ার ইজ এ ওয়ে, দেয়ার ইজ এ ওয়ে, বাট ইট হ্যাজ টু বি স্পেসিফিক ফর দ্য ব্যাকটেরিয়াল ডিএনএ।
ওকে কল্পনা, আচ্ছা সৃজিতা, হ্যাঁ
ঠিক আছে, তুমি
আচ্ছা
মোটামুটি মানে
সৃজিতা has asked another question, আমি ওটা শর্টে বলছি, ও যেটা মানে what she has meant is that for curing influenza fever there are different drugs like Baloxavir, Marboxil, Zanamivir, etc. Why is it necessary to design different drugs for the same disease? Why can't just one drug cure a specific kind of a disease?
আচ্ছা, এটা করতে গেলে প্রথমে ব্যাক টু দা স্কোয়ার ওয়ান। আজকে একটা ডিজিজ যখন শুরু হচ্ছে তখন ড্রাগটা আমি কি করে বার করব না, আমার একটা টার্গেট আইডেন্টিফাই করতে হবে, যে টার্গেটটা হচ্ছে স্পেসিফিক ফর দা ভাইরাস হোক, ব্যাকটেরিয়া হোক, যাই হোক, ঠিক আছে। আজকে কি হলো, এই যেহেতু তোমার একটা ড্রাগ দুটো মানে এই যে মিউটেশন, কনস্ট্যান্টলি মিউটেশন হচ্ছে, ফাইটিং ফর দ্য সার্ভাইভাল অফ দ্য ফিটেস্ট, দ্যাট প্রিন্সিপল, একটা কনস্ট্যান্ট চেঞ্জ চলছে, এবং এই চেঞ্জ চলছে বলেই তুমি একটা ড্রাগ দিয়ে বেশি দিন তুমি এটা করতে পারবে না, তোমার একটা ড্রাগে হবে না। তোমার আরেকটা হচ্ছে, প্রত্যেকটি ইনফ্লুয়েঞ্জা ভাইরাসের ক্যারেক্টারিস্টিক্স কিন্তু একটু আলাদা। তুমি ইনফ্লুয়েঞ্জা আনলে ইনফ্লুয়েঞ্জার 8 রকম ইনফ্লুয়েঞ্জা ভাইরাস থাকতে পারে, হ্যাঁ, তুমি দেখবে তার মধ্যে খুব ব্রড ক্লাসিফিকেশন হচ্ছে আরএনএ ভাইরাস, রেট্রো ভাইরাস, ডিএনএ ভাইরাস, ঠিক আছে। তা প্রত্যেকটা ভাইরাসের চরিত্র আলাদা, প্রত্যেকটার অ্যাম্প্লিফিকেশন আলাদা, তাহলে সেম ড্রাগ তো করতে পারবে না কিছুতেই। প্রত্যেকের এন্ট্রি পয়েন্টও আলাদা, স্পাইক প্রোটিন প্রত্যেকের আলাদা, তাহলে তুমি এক ড্রাগ দিয়ে করবে কি করে? বুঝতে পারছো, কোন ডিজিজের ক্ষেত্রে তুমি সেম ড্রাগ ইউজ করতে পারো? অ্যাসপিরিন আজও ইউজ হচ্ছে, প্যারাসিটামল আজও ইউজ হচ্ছে, কারণ এগুলো ইনফেকশাস ডিজিজের জন্য নয়, এগুলো আমার বডির নিজের মেকানিজমের এগেনস্টে কাজ করছে। ইনফেকশাস ডিজিজের এগেনস্টে তুমি করতে পারছো, কারণ সেখানে বাইরের এজেন্ট তারা চেঞ্জ করে নিচ্ছে নিজেকে, ইউ আর কনস্ট্যান্টলি চেঞ্জিং, তাই না? সেই জন্য এটা একটা ড্রাগ দিয়ে এটা হবে না, বেশি দিন তুমি করতে পারবে না। আর ডিফারেন্ট ফ্লুজ আর ডিফারেন্ট ভ্যারাইটিস, কারণ কোনোটাকে বলে রেট্রো ভাইরাস, কোনোটাকে বলে আরএনএ ভাইরাস, কোনোটাকে বলে ডিএনএ ভাইরাস। ইফ ইউ ওয়ান্ট টু নো মোর অর এনি ইন্টারেস্ট, আচ্ছা আমি একটা বলে দিচ্ছি, আমার ইমেলে যদি কোনো কোশ্চেন থাকে ইউ ক্যান অলওয়েজ কন্ট্যাক্ট মি ওভার ইমেল।
ভেরি গুড, তাহলে তো তোমাদের অনেক কোয়েশ্চেনের আনসার পাবে, বাট স্যার ফ্রম দ্য রেসপন্স obtained from the students it's very clear that they have been extremely enthused, and maybe I mean we are very happy to know that we have many promising budding researchers
Yes.
waiting to this is the purpose of carrying out this kind of webinars or seminars, so
Acha,
that
let me,
the students are
I think hopefully it was okay.
no, it was excellent, and from the response I think that you are also a bit satisfied that our students have
yes,
taken
definitely.
so much.
I think they ask very pertinent questions. And what I want to say, finish the last line, is that whatever I have taught belong to chemistry. So what we tell about biology today, when we see at the molecular level, it becomes chemistry. So we should be proud to be chemists. And
Absolutely.
chemistry is the central science. Unless you can draw structures, draw mechanism, you cannot overcome this resistance, everything.
Absolutely.
See, that we have to again reclaim our position, that chemistry is the central science.
Thank you so much,
And we
sir.
should be proud. Yeah, thank you very much.
Thank you so much. Just my formal vote of thanks after this such a nice and interactive and illuminating lecture. So on behalf of Lady Brabourne College, it is my honor and privilege to extend our sincere thanks to the illustrious academician, Professor Amit Basak, for sparing time from his busy schedule to join us in this virtual platform and deliver such a simple, lucid, and absolutely engrossing, excellent lecture, which I believe that has enriched not only the student, but enriched all of us extremely. My thanks also goes to our respected Madam Principal, Professor Shuli Sharkar, for her constant encouragement and support, not only in organizing this webinar, but in all our academic endeavors. Our thanks goes to the Department of Biotechnology, Government of India, for according us the star status, DBT star status, and providing grants for carrying out various activities such as seminars, workshops, projects, outreach programs, industry, and laboratory visits for the benefit of our students. And with great pleasure, I extend my thanks to all the participating faculty members, scholars from outside the institution, and those belonging to our institution as well. Last but not the least, thank you, our dear students, for participating so actively in this webinar and for your enthusiasm and for your wholehearted support. The technical support team has done a good job and deserves a big thanks. Sir, we sincerely look forward to have you in our college auditorium now, since the COVID is a bit hopefully in a declining.
Yes, I will always be. Yeah.
Status,
Yes.
And so our students will get an excellent opportunity to interact with an illustrious scientist like you face to face, and they must be having many more questions.
Yeah.
And many more queries to be answered.
Just one point to the students, that if you are interested, there is a course in the YouTube I have given, Organic Chemistry in Biology and Drug Design.
Organic chemistry, could you repeat, sir?
Organic chemistry in Biology and Drug Development. I think that is the name of the course. So YouTube, there are 63 lectures.
Biology and Drug Development.
Yes,
Okay.
63 lecture series there.
Okay.
Because my aim now is to reclaim our position as chemists. That biology is nothing but chemistry at the molecular level.
Thank you. Thank you so much. Thank you all for making this webinar a grand success, and wish you all good health, spirit.
Yeah, I
and
wish
good
you
spirit.
all also the
Thank
same.
you so very much.
Thank you.
Thank you.
Okay.
Okay, sir. Good night. Thank you.
Good night.